So welcome everyone to COVID Science Solutions and Success Stories. And without further ado, we're going to pass it on to our moderator with one quick introduction. But I did want to thank the committee that was putting this series together. I'm so happy to have had this series come to fruition. So hoping you can join us tomorrow for the solutions piece based on the hierarchy of controls. Today is the science piece, which I'll hand over shortly. And then there's a plea also for us to look forward to you, all the participants, for part 2 of this series, which will include success stories from all sectors around this pandemic that we're dealing with. We have a few people whose audio is not working, so I will pause a moment for Tony if you can help with that, and go ahead and pass it to one of the committee participants. We have Val, Kevin, who you'll hear from tomorrow, and Raymond Tellier, who's moderating today. And I wanted to thank very much Alec Farquhar, who will introduce Raymond now. Alec was director of MOL Occupational Health and Safety branch for several years just after SARS, which meant that he played a role in the implementation of the Campbell recommendations, and also worked with the late Dr. Sheila Basser, Provincial Medical Officer of Health, and Dr. Leon Genesov, then the MOL provincial physician, on pandemic preparedness. Following that, Alec was managing director of OCAO and then finished his career as director of the Office of the Worker Advisor. So Alec, thank you. And if you don't mind taking the torch and introducing Raymond, it would be appreciated.
Thank you so much, Kimberly. And thank you, OCAO, for hosting this vital series of webinars at such an important time. I'm very pleased to introduce Dr. Ray Tellier. Just a tiny bit more about my background. I'm a health and safety person, and really it was after SARS that I came into contact with the infectious disease world and some of the wonderful people working within it, such as Ray Tellier and the folks that you're going to be hearing from today. And of course, in that meeting of worlds, people speak different languages. People have different perspectives. We're at a moment where there are differing perspectives on the way that COVID-19 is transmitted. And very, very happy to have such a distinguished group today. And in terms of Ray, Ray and I didn't know each other. I reached out to him several weeks ago, knowing from his reputation that he'd find a way to bring the discussion to a very good point where we hear different perspectives and learn from each other. So just a little bit of background on Ray himself. Ray's an associate professor in the Department of Microbiology and Immunology in the Department of Medicine at McGill University Health Center. Ray's got a tremendous range of experience way beyond COVID-19, including areas such as coronaviruses, influenza and flu, hepatitis, respiratory medicine, bloodborne infections, viral infections of the immunodeficient, diagnostic virology, molecular microbiology, and medical microbiology. And this will give you an idea of the range of expertise he brings. And of course, this has allowed him to get to know some of the folks that'll be presenting today and understand their perspectives and how best to bring them together. So without further ado, thank you, Ray, for serving as our moderator. And along with everyone else, I'm looking forward to a very stimulating and important discussion today.
Thank you very much for this kind introduction. So, hello, can you hear me?
Yes.
Yes.
Okay, thank you very much for the kind introduction. So my name is Raymond Tellier. I'm an associate professor in the Department of Medicine of McGill University, and it is my great pleasure today to be the facilitator for this webinar. As we are still in the beginning of a global and accelerating pandemic caused by an emerging coronavirus, it is a good time, I think, to take stock of the state of knowledge about COVID-19 transmission, to identify gaps and uncertainty, as well as areas that are left unattended. Pending the availability of a vaccine, the only way that we have to control this pandemic is going to be through blocking the different routes of transmission of the virus. And so we are very fortunate today to have with us three world-renowned experts in this field who will share with us their knowledge and their assessment of the situation. Following the three presentations, which will be 20 minutes each, there will be a question period of about 25 minutes where questions can be addressed to any of the presenters. And so without delay, it is my great honor to introduce our first speaker today, Dr. Lindsay Marr. Dr. Marr received a bachelor degree in engineering from Harvard College. She received a PhD in civil and environmental engineering at the University of California at Berkeley and did postdoctoral studies at the Massachusetts Institute of Technology. She is currently the Charles P. Lunsford Professor of Civil and Environmental Engineering at Virginia Tech. She has been conducting and publishing several studies on airborne transmission of infectious agents, including respiratory virus. Dr. Ma, please.
Thank you for the introduction. Let me start sharing my slides. All right, could somebody let me know if you're able to see that? Cover slide, title slide?
Yeah, we can see it.
Great. So I'll be talking about SARS coronavirus 2 and droplets and aerosols. It's a real pleasure to be here today because Raymond's papers are some of the first I ran across when I first started getting into this field and asking questions about how transmission actually occurred. It was initially for the flu, but of course, those same principles apply for other respiratory viruses. So today I will be talking about different modes of transmission to get everyone on the same page. It's a bit of an introduction. And then I'll talk about sizes of viruses and the droplets and aerosols that carry them and why those are so important. We'll briefly touch upon evaporation and transport of the droplets and aerosols. How far do they go? And then I'll share with you some of the recent air sampling results for SARS-CoV-2 and some of the challenges that are inherent to sampling in the air. So the traditional definitions of modes of transmission can be broken up into four main modes. Sometimes people collapse these into two, but direct contact we're familiar with. That's shaking hands, of course, with someone, for example. Indirect contact involves touching an object that becomes contaminated and transferring the virus that way through, we call those objects fomites. And then there's what's called large droplet spray transmission, which occurs if you're close to someone and they cough or sneeze in your face, and the large droplets fly like mini cannonballs and hit you perhaps right in the eyes or the nostrils or the mouth, causing infection. And then finally, there's there's the recent debate about the importance of airborne or aerosol transmission, where an infected person releases virus into the air and aerosols that are small enough to remain floating around in air for a while that other people can then inhale. So the large droplet route is traditionally defined as for droplet sizes larger than 5 microns. And for reference, human hair is about 70 or 100 microns, so we're about 10 times smaller than that. And happening at close range only, less than 1 or 2 meters. Aerosol transmission has been defined as involving droplets, microscopic droplets, or we call them aerosols, smaller than 5 microns, and happening mainly at long distances or greater than 2 microns. The origin of the 5 to 10 micron cutoff is not clear. Most references point back to a paper from 1934 by Wells, but his paper actually says nothing about that. The cutoff is not supported by modern aerosol science, and this distinction, I think, has hampered our understanding of transmission. And I'll show you why here. So when you're in close contact, for example, person A is infected and person B is close to them, person A, either through just breathing or talking, coughing and sneezing, we know that breathing and talking also generate droplets and aerosols. So person B in this case is being exposed to the large droplets in red, which could fly through the air and land directly on their body. But there's also the even more of the blue ones, which are the smaller aerosols. And obviously the person has high exposure to those and could be breathing in a cloud of those, while person C, who's farther away, has no exposure. So when you're in close contact, transmission can occur both by droplet spray and by inhalation of aerosols. Later in time, as that plume of droplets spreads out, you could imagine a cigarette smoke plume spreading out in a room. The red ones, or the larger droplets, eventually fall to the ground, whereas the blue ones are able to spread farther in the air. And eventually, over longer periods of time, they can fill the air in the room and build up over time, such that person C, who's farther away, can be exposed through inhalation of these aerosols. This is why people are recommending ventilation, good ventilation of indoor settings, in order to eliminate the buildup of, or reduce the opportunity for the buildup of aerosols, because then you remove those, replace them with outdoor air, which presumably has little to no virus in it. There's been discussion of face coverings for source control, and they work for both droplets and aerosols. So if we have someone who puts on a face covering, it greatly reduces the amount of droplets and aerosols they release into the air, maybe not to zero, but even if they reduce it by half, then the person's exposure would be reduced by, the other person's exposure would be reduced by half. There's been some debate, as I mentioned, over what the exact cutoff size is for droplets versus aerosols. That cutoff is artificial. You should really just think of aerosols not as this mysterious, unseen thing, but as really a droplet that's just very, very small, microscopic, and too small to see. And I think rather than focusing so much on a certain size, we should be thinking about the route of exposure when we're distinguishing between these two. So I'd like to introduce the idea of droplets as being something where transmission occurs by contact, where the droplets land on the body, particularly the eyes, nostrils, or mouth, or land elsewhere in the body and then are transferred into the respiratory system. And aerosols generate exposure where you inhale them into the respiratory system. So we've seen that transmission by inhalation of aerosols can happen at short and long ranges. And the next question I'll address is, why is size so important? Well, let's talk first about the size of the virus. So SARS-CoV-2 is about 1.12 microns in size, similar in size to influenza virus. They're both enveloped. And so once they're out in the environment, they're going to be transported similarly. And we'll see that it's the droplet size itself that matters. Just for reference, we talk about air pollution and particles in the air. We talk about PM10, for example, shown in blue there, which is 10 microns. And then we talk about fine particulate matter, where we define the cutoff at 2.5 microns. And those are even more closely linked to adverse effects on health. Talking about a virus that is much smaller even than fine particulate matter. But the size of the virus itself, it turns out, doesn't really matter that much, because when the viruses get into the air, they are not naked. So even if we have a flu virus here, for example, that's 0.1 microns in size, it's released in a droplet of respiratory fluid. And that droplet can range widely in size from smaller than 0.2 microns up to 100 microns, or even larger than that. And the size is very important because this determines how far they can travel in the air and where they deposit in the respiratory system. And Dr. Milton will get more into details about where they deposit. Humidity is important because it affects both the size and the chemistry of the droplet, which is the environment, the microenvironment of the virus. So when the droplet is first released from the respiratory tract, it's at very high relative humidity conditions, right? Nearly saturation or 100% relative humidity. And I've shown the droplet here zoomed in so that we can see that it's not just water around the virus, but there's also salts and proteins too, and other components of respiratory fluid. In fact, there's almost 1 million times more salt and protein than there is of virus in terms of mass. So this is not to scale. The virus should be minuscule and really too small to see compared to the salt and protein here. So once that droplet hits ambient air, which typically is not at 100% humidity, it's going to evaporate. The smaller ones will evaporate faster. And at medium relative humidity conditions, such as we see in the spring or fall in temperate regions outdoors, we're going to get loss of a lot of the water, maybe most of the water. And you can see that the salt and protein, though, remain. So we still have the same amount of salt and protein because those cannot evaporate into air. They're still there. And then finally, at low relative humidity conditions, below about 40% or so, all of the water can evaporate, leaving behind just this solid residue of components of respiratory fluid along with the virus. So the initial size of the droplet's important, and also its final size, which depends on the humidity. And this is why, because the settling velocity, or kind of how fast those droplets fall to the ground, is a strong function of size. And so this figure shows you the diameter of the droplet on the x-axis, ranging from 0 to 50 microns. And the y-axis shows how long it takes for this droplet to settle from a height of 1.5 meters. And this calculation is based just on physics. I've shown the equation there. It's a function of g gravitational acceleration, and then also the particle diameter squared. And you can see droplets smaller than 10 microns can remain suspended for many minutes to hours. And if I highlight 5 microns, which is, remember, that cutoff between traditionally what we've called droplet and aerosol transmission, a 5 micron droplet actually takes half an hour to settle from a height of 1 meter. And to me, that is not settling immediately to the ground. This is something that poses a risk for inhalation exposure. Furthermore, these droplets can travel more than 1 or 2 meters. This graph shows the position of droplets released from a height of 1 meter. So on the left-hand side, all the up at a height of 1 meter on the far left at a distance of 0, that's where we release the droplet. And then the line shows the trajectory, or how far that droplet can travel under different conditions. On the left-hand graph, it shows a low indoor air velocity, and on the right hand, it shows a high indoor air velocity. So the air in a room is never perfectly still. It's always moving around a little bit. And I should also add that this is kind of after the initial kind of jet comes out of the mouth from talking or coughing or sneezing, because those droplets are going to slow down, and then now they're being carried by any currents in the indoor air. And so you can see that a 5 micron droplet can easily travel in the left-hand side at low indoor air velocity over 60 meters, well more than 1 to 2 meters. And even even a 20 micron droplet is going to travel almost 5 meters. That's the path shown in red. So droplets much larger than 5 microns can remain airborne and can travel quite far. And those distances are even extended further when we have higher indoor air velocities. So we've seen that viruses and droplets and aerosols larger than 5 to 10 microns remain in the air for many minutes and can travel farther than 1 to 2 meters. And now I'll get to the pressing question of our time, which is, what do we know about SARS coronavirus 2 in droplets and aerosols? So first, I need to go over a couple of different ways that we detect virus in air, because I'll be talking about some of those measurements. The first is the kind of total virus, or actually the number of genome copies, or number of gene copies more specifically, which are determined by molecular techniques, quantitative polymerase chain reaction, or qPCR. And this reflects the number of viruses that have intact DNA or RNA, depending on the type of virus it is, the number of intact, I guess, genes, because you're using a certain sequence. You're looking for a certain sequence in the genome. This measure, though, does not indicate whether the virus is infectious or not. So, for example, when flu viruses are made, only about 1% of them are initially infectious. The second method we use to detect viruses is to measure their infectivity, or their ability to actually infect cells, and we grow them or culture them or cultivate them on the host cells, and we count the number of plaque-forming units that form. So the picture on the right-hand side shows this lawn of purple bacteria, where the little clear spots are plaques, or holes where the virus has multiplied and killed off the bacteria. And you can see that, let's see, I got an annotation request. Let me just decline that for now. The left-hand side shows no plaques, and the right-hand side shows lots of plaques, because those are different dilutions of the virus that they put onto the bacteria. So we call these plaque-forming units, or sometimes focus-forming units, which are related. You'll also see the term TCID50, which is the median tissue culture infectious dose, or the concentration of virus at which half of the cells are infected after being exposed to the sample. So just as we have two different methods of detecting virus, kind of total gene copies by qPCR, and then infectivity by some kind of culture-based assay. Here's what we've seen so far for SARS coronavirus 2. This is from measurements in a hospital in Singapore. And the, let's see, these different numbers show where they collected samples in the patient's room. And they found that, as shown in the upper right-hand corner, surface samples were positive at numerous furniture locations, light switches, in the toilet, the door handle, the window, and the air outlet fans. The air outlet fans are shown by number 15 over on the left-hand side by the patient's bed, at the head of the bed. And it's unlikely, extremely unlikely, that the patient would have touched the air outlet fan. And so we presume that that got there by depositing there, or kind of sticking onto the air outlet fan, because the virus was traveling through the air. Now, there were also negative samples that were found where they did not find virus. They collected a sample, did not find virus, including the control panel on the bed, the outer rim of the sink in the bathroom, and the air. So that seems a little bit contradictory. It seems contradictory, because they found virus on the air outlet fan, but they did not find it in the air. We'll discuss why that might be here in a couple of slides. And so here I've combined, these are some measurements, additional measurements of viral RNA in hospitals by qPCR. A further follow-up study in Singapore resolved these by size, which, as I showed before, is very important for understanding how far the viruses can travel in air and how long they might stay in the air. And they measured it in 3 different rooms. So in one of the rooms, room 1, they didn't find anything in air. In the second room, they did find virus in air, and they found slightly more in the 1 to 4 micron size range, and a little bit less in the greater than 4 microns. And in room 3, they found similar amounts. So we're looking at the order of 1,000 copies of the gene copies per cubic meter of air. So in Nebraska, the biocontainment unit and national quarantine unit, they found numbers. This is kind of total in air, not segregated by size, but they found numbers from 2,000 to 3,000 copies per cubic meter. Now, both these hospitals have high air exchange rates, or high ventilation rates in their rooms of, I think, 12 to 15 air changes per hour. Measurements in a hospital in Wuhan also detected SARS coronavirus 2 RNA in, excuse me, in aerosols, what we would call aerosols, or smaller than 5 microns. So this, the left-hand side shows concentrations in the protective apparel removal room of up to 40 gene copies per cubic meter. And then you can see the middle figure shows it in the protective apparel room also, a different one. And this shows more virus across, well, sorry, these absolute numbers here are lower. The Y scale is lower. But you can see that virus is found across the whole range of sizes that they measured. 2.5 I'll highlight because that's kind of where we cut off fine particulate matter for air pollution. But you can see there's plenty of virus in these aerosols that are smaller than 5 microns, and you can see that in the right panel too in the medical staff's office. We know that virus laboratory experiments have shown that the virus survives in aerosols, so the RNA has been detected. The question is whether, if it was infectious, could it survive? There was a study published a couple of months ago where they looked at survival in aerosols, and they found a half-life of SARS coronavirus 2 right around 1 hour, which is similar to the half-life of SARS coronavirus 1. So clearly the virus can survive, at least under these conditions. And people have criticized these studies as saying, well, it's a laboratory condition, it's an artificial aerosolization technique. My group has done these types of studies. This type of aerosolization technique, I would say, is harsher than you would have in natural conditions. And then also they used a cell culture medium, and we have found that virus survives even better when it's supplemented with real respiratory secretions. So I would say this is probably a minimum half-life. And there was another study recently published where they showed that the virus in aerosols, fine aerosols, very small ones, survived for many hours. This experiment ran out to 16 hours. So here are some of the challenges of sampling viruses in air. There are very low concentrations. In the hospital, we saw 1,000 viral copies or RNA copies per cubic meter of air. With this virus, it seems like you probably need about 10,000 copies to get one PFU or one TCID50. This means that you need 10 cubic meters to sample 10 cubic meters of air. And ideally, you want size separation of the viruses too, so that you can figure out how far can they go. So a couple of the sampling techniques shown on the left are able to segregate particles by size or droplets by size, but they have low flow rates. And it would take 20 to 40 hours of sampling to get our 10 cubic meters, which would only give us one plaque-forming unit, one infectious virus maybe. The middle figure shows a different sampling device, which can run at a much higher flow rate, but the filter, which is kind of the circular thing on the end, is fairly large, and you have to elute that and then extract it. And there's inevitably losses along the way. It's hard because you want to concentrate that down to a very small volume in order to do the qPCR analysis. A further challenge is that just during the sampling itself, there can be loss of infectivity during sampling. So, it's true we have not seen infectious virus grown from air samples yet, but I think there are reasons why we haven't. So there are still major unknowns, of course. Which transmission route is dominant? This is the million-dollar question. Is it direct contact or fomites or inhalation of aerosol, of contact with droplets? The second major big question is how much virus is released and what size droplet aerosol or aerosols at different stages of infection. And then finally, how are viruses inactivated in air and on surfaces? Finally, I'd like to thank all of my collaborators on this. There's faculty at Virginia Tech and the University of Pittsburgh, postdocs and graduate students who have worked on this over the years. And now I'll turn it over to the next speaker, and I'll be happy to take questions, I think, during the panel discussion at the end. Thank you.
Thank you very much, Dr. Ma, for this illuminating presentation. It is also my great honor and privilege to introduce a former colleague of mine at the University of Calgary and our second presenter, Dr. John Connelly, who is a physician, a specialist in infectious disease. He is currently professor at the Cummings School of Medicine at the University of Calgary, where he not only practices as a consultant in clinical infectious disease, but also attends to infection control. Dr. Connelly is a world-renowned researcher in the fields of antimicrobial resistance and stewardship in infection control and novel innovation in healthcare. He, among many other activities, he is also the chair of the World Health Organization Infection Prevention and Control Research Innovation Advisory Group. And so it is with great pleasure that I leave the field to Dr. Connelly, and I look forward to his presentation.
Thank you very much, Raymond. Are you able to hear me all right? Just do a sound check.
Yes, it sounds good.
Okay. Thanks very much for the opportunity to present to this group. And I'm going to try to take you over the next 25 minutes or 20 to 25 minutes, some of the issues related to modes of transmission and IPC measures that can be used to mitigate it. I'll just begin with an acknowledgement, territorial acknowledgement, and I'm going to acknowledge the traditional territories of the Blackfoot and Treaty 7 peoples. It includes the Siksika, Papunai, Kenai, Tsuut'ina, and Stoney Nakoda First Nations. And also Calgary is home to the Métis Nation of Alberta, Region 3. So just to make that territorial acknowledgement for the First Nations peoples. And as far as disclosures and acknowledgements, I have one item to bring up. I was the local site investigator for Staph aureus vaccine trial in instrumented spinal fusion that was funded by Pfizer, but all of the money went to the University of Calgary. I received no personal remuneration. I also want to acknowledge the Li Ka-shing Institute of Virology and the Provincial Laboratory of Alberta, with whom we've been interacting for some cultivatability studies, which I will be releasing to you for the first time. No one else has seen this data, so I'm going to be presenting some original data to you. One thing I wanted to just mention right up front is that this is the first pandemic in which we've had a large amount of social media involvement. It's the first modern age in which we had H1N1 in 2009, but cell phones and Twitter and TikTok, they're just so much more prevalent now. And we have a lot of papers that are published online before they're peer-reviewed. We have preprints that are coming up, and the science may be sometimes correct, but media can jump to premature conclusions. Some aspects will never be published. We've got a race to the top. Who can be first? Who can be fast? We often see top quality peer-reviewed journals. You're probably aware that there have been major retractions in both the New England Journal of Medicine and the Lancet. There are more that are pending now as well. And so I advise everyone to be very careful and use your own critical appraisal skills and use a good element of common sense in looking at some of the papers that are coming out. Here's one that came out in PNAS. They have this peculiar system where you can call your friends up to do peer reviews. It's a completely corrupt and biased peer review system within the academy. This is in the United States. And a group of 50 epidemiologists, I think they could have had 500 or 5,000 easily, pointing out the egregious errors where they were commenting that there's airborne transmission in New York City by putting a couple of correlation coefficients with stay at home and then face mask covering, and came to the conclusion that airborne transmission was the dominant route. Many people have called for this to be retracted. It's full of egregious errors and many false statements. Many of us in the infectious disease epidemiology are going to use this in our teaching now for graduate students and medical students of a classic example of an epidemiologic ecologic fallacy. So that's one good thing that came out of this, is we can now use this as a mechanism to be able to show how data is manipulated to be able to come up with a very erroneous conclusion. Some basic viral facts about SARS coronavirus 2 causing COVID-19 infection. And just going to get rid of this item here. It's a single-stranded RNA virus. It's in the beta coronavirus family. We know that it's one of a number of coronaviruses that can cause infection. We have the human coronavirus we've known about for many years. And then also the SARS coronavirus 1 and the MERS coronavirus as well. And it's a respiratory virus, and we traditionally think of these as the predominant routes of droplet and contact transmission, and I'll touch on that later. One thing that we have seen with this virus is it seems to be more contagious than SARS coronavirus 1 and MERS, a little bit different in that those latter viruses would ramp up their viral loads later in the illness. With this particular virus, we see an almost immediate ramp up of fairly significant viral loads. And we also see that there's a much higher affinity for the ACE2 receptor. This is prevalent in respiratory epithelial lining cells. You have the angiotensin converting enzyme, and it's got much greater avidity than SARS coronavirus 1. So one thing that's of interest is whether there could be a lower inoculum to be able to elicit invasive infection with this virus than with its companion viruses in the beta coronavirus family. My slide is not advancing. This happens sometimes. One moment. There we go. As far as the origins of the virus, it likely originated from the Asian bats. Some of you may be aware of this. There's some question about whether there's an intermediate host with the pangolin or the Asian anteater. We know that with MERS coronavirus that the camels were the intermediary host. One thing that we've seen is that the virus does not appear to be mutating significantly, so it is possible a vaccine will be available. There are 149 vaccine candidates. 17 are now being used in early clinical trials, phase 1 and phase 2. So hopefully we will see something. And there is a group within the WHO through the Solidarity vaccine trial that they're looking to be able to provide vaccine availability. So hopefully we'll see the end of this with the development of a vaccine. The one important item to note is that no one is really immune from this virus. It's natural history in humans. I'm not going to spend a lot of time on this. We know that there are a certain proportion of infections that may be asymptomatic. We know that there may be pre-symptomatic individuals who may transmit. There's been some recent studies that have shown higher rates of transmission from those with more severe clinical illness. So those that have a sputum production, for example, who have a lot of cough and are quite symptomatic. And the variation in the well-done studies suggest that asymptomatic transmission is only in the range of 0 to 6%, but that there's been a stepwise increase in a couple of very well-done, elegant studies where there's been increasing proportions of individuals in a stepwise fashion with a dose-response curve, which scientifically makes good sense as well. We know that most of those that are going to be infected do so within about 10 to 11 days, and 80% get self-limited illness, 15% have severe illness, and 5% go on to intensive care and ventilatory requirements, which is very unfortunate because if you end up on a ventilator, the mortality is in the range of the case fatality rate in about 70%. One of the things we don't understand well is what are some of the drivers of individuals who develop more severe illness. We do know there are a few factors with comorbid conditions, obesity, and there are certain people that seem to have a predilection, such as those with African American heritage. But the genetic makeup of individuals as to why some develop very severe cytokine storm and or overwhelming viral pneumonitis are just not well understood at this point. Here's the virus here and the coronavirus. You can see where COVID comes from, just to remind you. And here's the transmission routes. The prior speaker went over this, so I'll just briefly mention we traditionally think about the infected individual who may produce droplets, airborne, and then we have direct contact and indirect contact. And these are the routes. But one thing that's important is that we know that droplet and airborne seems to create the most debate. It's important to recognize that I think we recognize from an infectious disease epidemiologic perspective that there's a continuum from droplet to airborne routes. It's not an all or none phenomenon. And I think that's important for people to be able to realize is that this is some type of a continuum with respect to the variety of sizes that are expelled from the human airways during coughing, sneezing, talking, and aerosol generating medical procedures. And it's very complex. I think Dr. Mara presented the complexities in looking at this. And I'm not an expert in the aerobiology, but there are many, many factors that have to be looked at. They were elucidated previously, the particle size, the distance they can be propelled, the force that's generated by the individual or the procedure that's being done. The particles may or may not contain infectious agents. In speaking to my colleagues in virology, I'm working with the Institute of Virology. They have probably over 100 person-years of experience in basic core virology. They're basic scientists. And they explained to me that you have variable amounts of virus that may or may not be present. Some may be partially desiccated, some are defective. Replication competent does not equal infection competent. And so you need to have replication competent and infection competent virus to be able to transmit. So it's a very complex scenario to be able to look at. And you have to look at the concentration of the particles and then the concentration of virus that may be present It's affected by humidity, evaporation levels, the setting, the velocity, direction of airflow, temperature, crowding, many environmental factors. So very complex scenario, and why I think it's very difficult to be able to find cultivatable, replication competent and infection competent virus in this particular setting with SARS coronavirus 2. We also have opportunistic airborne, which can occur where you've got settings in which aerosol generating procedures are in which you can have opportunistic airborne. It's not something that would be traditionally thought of in that capacity. When we look at this from an epidemiologic and an infectious disease perspective, we have to have exposure to microorganisms. We have to have the recognition that not all exposures lead to transmission or invasive infection, and that you have exposure that occurs when a host comes into contact with a contaminated environment from whatever reason, inanimate objects, droplets that may contain virus. And then you've got to have a susceptible host. You've got to have the presence of host receptors. You've got to have receptivity of those host receptors. You have to have sufficient inoculum of replication and infection competent virus. You need to have a very healthy set of viruses to be able to inoculate this individual and also have virulence. And then you also have to look at the hierarchy of controls that might be in place, particularly in a hospital setting, that might retard the ability of a virus to be able to inoculate and cause invasive infection in the host. So I just want to impart to you that it's a very complex set of scenarios that have to be looked at from the standpoint of having infectious virus, which is replication and infection competent, to be able to deliver a sufficient inoculum to cause infection in a susceptible host. So a lot of very complex factors that come into play. With respect to SARS coronavirus 2, just to briefly review where we have cultivatable virus, BAL samples are loaded with the cultivatable virus that's been shown in multiple studies, from Zoo, the original study, right up to Van Kampen, that's just been published by the Dutch group. It's on the preprint server, where they looked at the largest subset of 129 patients to date. We know that there are multiple reports of the surrogate in nasopharyngeal, throat swabs, and saliva, looking at RNA fragments. And it's important to recognize that it does not always correlate with the presence of cultivatable virus, and I'll touch on that in a moment. We have RNA fragments that represent dead virus, which can be found in patients now we're discovering up to 6 to 8 weeks after infection. There's no natural RNAs, and so as a result, you can find detectable RNA fragments for very long periods in the absence of cultivatable virus. Most of the studies, and there have been several very well done, elegant studies now by core virology laboratories, Bullard and NML in Canada, also the Van Kampen in the Netherlands, and one other major paper that has shown a virus by Wolfel that was in Nature, that most of the cultivatable virus with this particular virus is gone by day 7 to day 8. It's pretty rare. In the Van Kampen study, they found in some ICU patients about day 10, 11, they found some cultivatable virus, but it does rapidly fall off, and I'll explain why that is in a moment. Also that RT-PCR positivity has been found in serum. There are now 3 studies where cultivatable virus has been found in stool, and there's been a recent publication to show, I think in Cell, that enterocytes are a capable cell line that can allow cultivatable virus. So people are now beginning to question whether fecal-oral transmission could be a route of transmission. Cultivatable virus has also been found in urine recently in another study. So, and these are important studies. We've been doing some work recently to look at patients in our own institution, and we transport up to the level 3 laboratory in Edmonton. And we've been using multiple culture techniques using minimal essential media and universal transport media. They use a Aerosol line and are very capable of growing viruses. And what we found is we've used a number of different collection techniques and we've been looking at patients from day 1 post onset of symptoms up to greater than 10 days. Of the 11 patients we found to date, the virus, cultivatability, and in quite significant titers, which I'll share with you in a moment, we find quite readily within a mean of 4 days post onset. For the remainder of the patients who have a mean onset, post-symptom onset of 10.3 days, they're almost all culture. In fact, they are all culture negative. So we have a great deal of difficulty in finding them. We've been culturing the environment. We found nasal prongs that are lying on the bed or sitting next to the patient that are used for oxygen delivery. And we found anywhere from 10 to the 1 up to 10 to the 2 plaque forming units per ml off nasal prongs, just wiping them with a gauze dipped in essential media, and not too difficult to find. We found saliva specimens to date. 4 of 6 are positive, anywhere from 8 times 10 to the 1 to 2.7 times 10 to the 3 plaque forming units. And then people just spit saliva into a cup and were transported up. And remember, these specimens are stored overnight in Calgary, and then they're sent 300 kilometers up the road via transport system with the provincial laboratory. So they do sit for 24 hours, and yet we're still finding specimens that are positive. We found a sputum sample. This is one that I collected many of these myself, and I found one sputum sample sitting on the gown of a patient in a room. It had 1.25 times 10 to the 6. That's a massive amount of virus, plaque forming units per ml, in a coughed up sputum specimen just lying on a gown. We found another sputum sample that a patient was spitting up at 4.5 times 10 to the 2. We found hand washing bags. We've just started doing this. We had someone who was about day 4 to 5 in illness. We had them blow their nose in a tissue. Then we had them cough into cupped hands. And we diluted it in 10 mLs, but we found basically 2,000 plaque forming units per ml, we estimated, in the hands of this individual. They just coughed into their hands. So for those that are saying that we don't find it in the environment, that's not our experience. We find quite significant quantities of virus that are both replication and infection competent. We've been using a unique system with cough bags. We basically went to Safeway and bought Ziploc bags. We hold them about 5 centimeters from patients. And we're now just doing this in patients who are within the first 5 days of illness. And in the cough bags, we find anywhere from 5.5 times 10 to the 2 up to 10 to the 3 plaque forming units per ml. We just find them cough into the bags. We can see a spittle in the bags macroscopically. I've done many of these at the bedside myself. We've also used techniques with Teflon sheets, and we can see spittle from the coughs, on the Teflon, and then we scrape it off with a spatula. That's been inferior, so we're just using the cough bags. We've also done multiple talk and singing bags. We have yet to find a positive specimen, but in 50% of them, we easily find quite significant amounts of virus in cough bags, a very simple technique where they cough into the bag. And we've also done settle plates at 1 and 2 meters. We have yet to find a single cultivatable virus in this setting in these patients. And as I mentioned, you're the first group that's seen this data. We continue to collect this, and we'll hope to expand the number of patients, but we're actually running out of patients now at this point, unfortunately. This is stuck again. Just give it a moment. These slides stick from time to time. I can't bring this forward for some reason. I may have to come out. Sorry about this. This sticks sometimes on WebEx for some reason, and none of my buttons are allowing me to advance my slides. There we go. Just to show you that the virus, and there have been several studies. This is just one showing that it can be found on many surfaces, glass, plastic, up to 4 days. This is under optimal condition. This is both MERS and SARS coronavirus. Another study showing the survival of the virus on stainless steel cardboard, plastic, again at 48 to 72 hours. And similarly, another more recent study in the Lancet where they found it living on plastic up to, again, 96 hours. So of all of the substances, plastic does seem to be the one in which we see the virus surviving the longest. This is the Wolfel study that I just wanted to show you. Day after symptoms, by day 10, they were able to show that there was no longer cultivatable virus in 10 healthy subjects, but yet they were finding RT-PCR positivity in many instances at 10 to the 3, 10 to the 4 log 10 copies per per swab. So we know that, in fact, you can find dead virus in these patients after about day 10, and that's been shown in 3 studies now. This is the Van Kampen study that shows, again, by about day 10, you can't cultivate virus very readily, and that probably correlates with the fact that you have neutralizing antibody titers that are being produced in these individuals, for the most part in healthy individuals, but also in some patients with multiple comorbidities, they're finding very similar findings. Just to remind you, and I'm going to wrap up in a moment, that in the previous SARS and MERS coronavirus setting, we were able to find that compliance with gloving and gowns, medical masks or N95s were applicable, was very important, and that if you had respiratory secretion contact, it was a major risk, eyes and mucous membranes for the development of SARS coronavirus 1. There were multiple reports, at least 7 accounted, where there was the presence of transmission even with N95 respirators, and again the importance of fomite control and careful hand hygiene. The predominant mode of transmission of SARS coronavirus 2 from a clinical epidemiologic perspective is through droplet and contact. It's consistent with the reproductive number of other contact droplet viruses. Some people have complained, oh, you can't compare measles and varicella to this virus, but many virologists say they are very different viruses. The joint emission in China showed the vast majority of the clusters were within families. That's now been reported in studies in Korea, Taiwan, and the US. So this has been corroborated in multiple other settings. And here's the reproduction number. You can see with most studies, the reproduction number falls in the 2.2 to 2.68 range. It was about 2.6 even in the early stages in both Korea and Italy. And there are multiple reports. I'm not going to go through all of this. I think the previous speaker talked about for and against airborne transmission. There have been multiple clinical studies of individuals who were cared for without the use of masks, but with careful hand hygiene and the use of gowns, gloves, where there is no evidence of transmission. There was the report of an air-conditioned restaurant in China where there was a distance between the tables that was only 1 meter. And I think it still remains to be seen whether there could have been transmission within that 1 meter. The aerosolized virus has been commented on that this is done with a powerful 3 or 6 jet nebulizers. It does not simulate the human cough condition or what we might see in most patients in a hospital. These are experimental studies, as was mentioned by the previous speaker. And then there are now 14 studies that I just pulled that up this morning, looking against or for the presence of airborne transmission. 8 have looked at surrogates with RNA and have found no RNA in air samples. 6 have found that, and that was just touched on in the previous study. Not a single study to date has found viable virus. I think it's going to be doubtful that they will. It would take a great deal of virus to be present in a normal hospital room setting to be able to find cultivatable virus. And I think in talking to my virology colleagues, they are very doubtful that one would be able to find healthy, replication competent and infection competent virus in air samples that would make this a very significant route of transmission of the virus. In Calgary, I'll just close up now. We had over 5,000 cases of SARS coronavirus, 168 inpatient admissions. We've carefully followed all of our 132 patients on the wards. We use the Public Health Agency of Canada, which is gowns, gloves, medical masks, and face shields or goggles for the care of these patients, and very careful hand hygiene. We've looked now over 5,544 continuous person hours of contact, which is roughly the equivalent of about 7 to 8 months. We've not had a single transmission event to a healthcare worker. So the recommendations that the Public Health Agency, which are based on the WHO, do work. We are quite confident in that and have been using this since the outbreak and have not seen a single transmission to a healthcare worker. I've collected most of those samples myself. I don't want to use anecdotes, but feel quite comfortable in my medical mask, goggles, gloves, gowns when I collect specimens from any of these patients. We've also recently worked up 5 symptomatic healthcare workers. Most of the CT values were in the 10 to 20 range. We had one as low as 10. We had close contact, less than 1 meter in 15 minutes, and we found no transmission events. We've just submitted that for publication. And there are now publications from Hong Kong, Italy, and Germany showing that there is no increased transmission risk with the use of medical masks as a component of the PPE that are used. And so just to finish up here, the principles of IPC strategies, you want to ensure triage, early recognition, apply standard precautions for all patients, use your droplet and contact transmission recommendations. So medical mask as a component for routine care, and then also N95 respirators for aerosol generating procedures, and implement administrative controls, and of course, careful disinfection of the environment and careful hand hygiene. And with that, I'll close. Thanks very much for your attention.
Thank you, Dr. Connelly. I'm moving now to our third presenter, Dr. Donald Milton. Dr. Milton earned his MD degree from Johns Hopkins and obtained a master's in occupational health and a doctorate in public health from Harvard University. He is currently professor of environmental and occupational health at the University of Maryland School of Public Health. Dr. Milton is a world-renowned researcher and expert in the field of infectious disease aerobiology, including infectious aerosol generated during infection by respiratory viruses. So it is a great pleasure to return the microphone to Dr. Milton. Thank you very much.
Let's see. Can you hear me?
Yes, we can.
Okay, so let's see. Share content. All right, and hopefully you are now seeing my screen and seeing the slideshow. So thank you. I appreciate this opportunity to present and talk about this important topic, trying to understand and control SARS-CoV-2 transmission. So my outline, I'm going to talk about the elusive pathway and what is an aerosol, and reprise a little bit of what Dr. Marr was saying earlier, and talk about humans as aerosol generators and how we sample breath, where SARS-CoV-2 receptors are located within the respiratory tract, and then talk a little bit about source control and environmental control. So this diagram is designed to illustrate the aerobiological pathway, that if a transmission is occurring via aerosols, what are we talking about? Where do these come from? So the upper, on the left side here, you see the upper respiratory tract can generate droplets mostly in the 1 micron and above sizes, whereas from the lower respiratory tract, we can generate very tiny particles when we breathe. When you shake out things like a gown that has been contaminated and create an aerosol, these are mostly going to be very large droplets. And if you have water sprays, they can be of a variety of sizes. And the reason we talk about sewage and water sources in this slide is because this figure was developed by an artist at the New England Journal of Medicine to accompany a active article that was published along with a paper about the Amoy Gardens Apartments outbreak in Hong Kong of SARS in 2003. once a droplet gets into the air, as you heard a little bit from Dr. Marr there, it is dynamic. It can desiccate, it could dry out, it can fall out of the air depending on its size, and we call that physical decay, or it can, in terms of infectiousness, biologically decay. And you saw some data about how long the SARS-CoV-2 virus can persist in an infectious state in an aerosol under laboratory conditions, which were actually designed to be relatively adverse for the virus, not permissive. So probably a worst case is that it can survive for an hour or so and perhaps longer. Then where the virus, the particle would land in your respiratory tract when you inhale it will depend on its size, with it larger droplets being retained in the nose, smaller ones penetrating beyond the larynx and into the tracheobronchial tree, and the finest ones down into the pulmonary region, although particles of about 0.3 microns are not retained very well at all anywhere in the respiratory tract. Very much in the same way that the lung is just acting as a filter, and reason why we test filters with 0.3 microns is because that's the size that doesn't collect very well. So there are two ways to define droplets and particles that can carry infections. There are the medical categories of calling them droplets or calling them aerosols. And typically, it's thought that droplets don't travel very far, and it's thought that mostly what they're doing is contaminating surfaces, such as the gown that we heard about from Dr. Connelly, and they're not considered airborne infection transmission, whereas aerosols, sometimes called droplet nuclei, are thought to be the desiccated remainder of droplets and be less than 5 microns, small enough to travel long distances and cause infection far from the source. And these are what are traditionally considered the only cause of airborne infection. On the other hand, many of you are probably familiar with the, given that this is sponsored by an occupational health organization, the industrial hygiene and exposure science classification of aerosols, where we talk about total inhalable aerosols, which can go up to quite large because the nose, the nostril can emit fairly large particles if they happen to be present in your breathing zone, such as sawdust or pollen. And we then talk about, but they don't get past the nose very well. So we call them total inhalable. And then we talk about thoracic, those that pass the larynx and get into the trachea, bronchi, but not very far beyond that because they run into something and get deposited. And finally, we talk about respirable aerosols, which are less than 10 and mostly less than 5 microns and penetrate deep into the lung. From an air pollution perspective, we tend to talk about particle matter less than 10 microns or less than 2.5 microns. And in a similar way with the less than 10 microns penetrating into the lungs, but not to the alveolar level, and PM2.5 penetrating much deeper. A lot of this is driven by this data here demonstrating where things deposit. And you can see that even when you're at 2 microns, where you have a lot of deposition in the alveolus, most of the stuff is still depositing in the nose and mouth. And the same is true in the tracheobronchial fraction. And we're not much concerned about these smaller particles here because the virus itself is around this size. And as Dr. Marr explained, if it were present in the air, it would be with other things that were present in the respiratory lining fluid. Now, Dr. Marr mentioned William Wells as having been attributed to the source of this idea that things less than 5 microns don't travel very far. In fact, he was quite clear that things less than 50 microns could dry out quickly to become things fairly small. But what he really showed was that things that were less than 5 microns were critically important for TB transmission. In this seminal experiment where he exposed an experimental animal, a rabbit in this case, to aerosols of TB in large coarse droplets and in fine droplet aerosols, and showed that in the coarse droplets, the animals did not become infected. The particles landed in the upper respiratory tract and were cleared away because the vulnerable cell in the case of TB is the alveolar macrophage. So if you don't deposit the bacillus deep in the lung, it doesn't cause infection readily. So turning to that's the source of this idea that less than 5 microns is what is important and is airborne, and that's where TB lives, okay? So what about people as a source of aerosols? What size aerosols do we generate? Well, this work by Lydia Marwaska really elucidated a understanding of airway reopening. People, when breathing, gradually have closure of airways, and especially if you exhale to low lung volumes, most of us will have some amount of airway closure. And when we take a deep breath or sigh, we will pop them open. And in the process, there's a film burst that creates little droplets that are sucked into the alveolus and exhaled on the next breath. And multiple labs have shown that if you have people exhale to residual volume and then inhale deeply, that on the next breath, they will increase their generation of very small aerosol particles in the under 5 micron and even under 1 micron range in the next exhalation. And Dr. Marwaska's lab did some really beautiful work looking at the size distribution of aerosols from breathing, from coughing, and talking, and showed that there are really 3 modes. There's a fine particle aerosol down around 1 micron that she calls the respirable mode, the breathing mode. There's a laryngeal mode and an oral mode, and that these overlap with the oral mode mostly being up in the relatively large size over 10 microns. Now these then can dry out, and as Wells showed and others some more recently, that particle droplets that start out in this size range up to about 50 to 80 or even 100 microns can dry out and become much smaller particles that remain suspended in air for a long time. And you don't need to get as small as 5 microns to travel a long distance, as Dr. Marr has shown. Also, this work from UC Davis more recently has looked at loudness of speaking and how much aerosol is being generated. And clearly, the louder one speaks, the more aerosol is generated. So where do the cells live that are the target for this virus? So we said Wells has shown back in the 1930s and 40s that it required deposition deep in the lung to infect with TB. What about this new SARS virus? So this is from a couple of recent papers looking at where are the receptors for the SARS virus, the ACE2 receptor that you may have heard about. And so this is looking at the histology without infection with the SARS-CoV-2 infection and with the SARS-CoV-1 infection. And you can see that there is very diffuse receptors in the bronchus, and there are scattered receptors in the lung, and there are even receptors in the conjunctiva. And in another paper by Xu et al., they showed that there's a lot of receptors in the mouth, on the tongue, on the floor of the mouth, the base of the tongue, and other sites within the mouth. So that basically the entire respiratory tract from the conjunctiva all the way down to the alveolus, there are receptors for this new virus. And there are a lot of them. So we are susceptible throughout this range. So the question is, how do you get infected? Well, you get virus most likely to one of these sites, but your GI tract is also susceptible. So if you eat or drink something contaminated with the virus, you can probably get infected that way as well. When this is another paper from Singapore, following on the one that looked at sampling for RNA and found the RNA on the exhaust vent in the hospital room. They went back with different samplers and a different arrangement of samplers around the patient's bed and sampled and were able to find virus in the air in the patient's bedroom, even though the airflow entered the room over the bed and was sucked out behind the bed so as to have a directional airflow and try to keep air away from the sides of the bed that would be contaminated by the patient. And this room had 12 air changes per hour. So compare that to in the average home, where the air changes are probably usually on the order of a half an air change an hour, and maybe even less in homes that have been sealed up tight for energy efficiency. So here you're seeing 2,000 copies per cubic meter of air in the greater than 4 micron, which would deposit in the bronchus or above, and 1,000 or so in the 1 to 4 micron, which would be in the respirable range and reach more deeply in the lung. To put these numbers in perspective, we have to remember that a person breathes 12 to 14 cubic meters of air a day. So this is the amount in one of those, potentially in one of those cubic meters in a very highly ventilated environment. So we can see that in such a highly ventilated environment, if you are there for shorter periods of time and are even wearing a surgical mask and cutting out 2/3 of the material coming in, that your exposure is going to be limited, but that if you're in a poorly ventilated environment, you may have much, much higher exposures to aerosols over a wide range of sizes of these aerosols. So I'm going to talk a little bit about some of the work that we've done in my lab. We were to look at coughing. We heard Dr. Connelly talk about having people cough into a plastic bag. This is a much more complicated way of doing this that was developed in the 1960s at the U.S. Army Institute for Infectious Diseases in Fort Detrick, Maryland. And this is a picture of this paper by Jerome and others and Vernon Knight, and I believe this may be a picture of Vernon Knight himself, coughing into a face seal against a large cone, which says Gesundheit on the side. And he's in a plastic booth that's being supplied with filtered air. And he has a weather balloon in here that inflates as he draws air samples out of this in order to collect on filters and impingers and other devices that are designed to collect the fine particles that you wouldn't find, for example, just coughing and then wiping the walls of the thing, because they would have not fallen out that quickly. Similarly, coughing into a plastic bag will capture the large droplets that are in the spittle, but the fine particle aerosols would escape the bag as soon as you took it away from your face. So you wouldn't find them there. We developed a device we call the Gesundheit 2 to recognize the pioneering work of Knight and colleagues. And because it uses a cone, although we turned it around the other way, and rather than having a static sample where one coughs into the cone, we have a dynamic sample where the person just breathes, talks, wears a surgical mask or not, while we sample the air, and we pull the air through a slit and have an impactor right about here in the throat of the cone that collects everything larger than 5 microns. And because of the capture velocity that we have here, a face velocity of the cone, if you're used to thinking about capture hoods, is such that we are able to entrain everything up to 80 microns with about 100% efficiency, and it begins to drop off a bit after that. And we then immediately add humidity to the air and then chill it rapidly to grow the size of smaller particles so that we can easily collect them in a liquid reservoir that's not shown down below. And this is able to then collect anything bigger than 0.05 microns and less than anything that gets past this 5 micron impactor up above. And we call them coarse and fine aerosols, again, following the categories that William Wells described many, many years ago. We, using this device, have been able to show that influenza virus is culturable in the breath of volunteers. Up until this, using a rather sophisticated approach like this, it was not possible to show that there was infectious virus in people's breath. But we were actually able to show that the average influenza case we recruited from the college clinic had about 1 infectious virus per minute in their breath. The virus is present in exhaled breath even without coughing. And over this range, our detection limit is somewhere around 10 to the 2 or 10 to the, a little under 10 to the 3. And we were able to culture virus from some of these cases in this group with no coughing at all. Now, granted that people who were coughing a lot were shedding more virus, but coughing wasn't the only way that they were shedding virus. This device was originally developed for the purpose of testing whether surgical masks work as source control. And so we looked at whether the RNA that we were collecting was knocked out by putting surgical masks on people. And in this first study, published in 2013, we had subjects wear a surgical mask for 30 minutes in the device, then cleaned up, brought them back in without a mask, and collected a paired sample so that we could really compare within persons, because you can see that the range of shedding between people is quite large. And we were able to knock out almost all of the virus in exhaled breath in the coarse particles, and a fraction of it here. This is a log scale, so it's dropping from 100 down to around 10. So we were all told, although there were more viruses in the fine particles, so that dominated it, all told, we were knocking out about 2/3 of the virus in exhaled breath using a surgical mask as source control. In a recent paper published in April in Nature Medicine, the same device was used in Hong Kong to study people with all sorts of respiratory illnesses, rhinovirus, influenza, and coronavirus. And it appeared that in the coronavirus cases, while the shedding was not very high for seasonal coronaviruses into aerosol, that it was there, and that the surgical mask appeared to eliminate it. Now, these weren't paired samples, so I would just have the caveat that this may not look quite as good when you run paired samples, but the data suggests that surgical mask as source control are important. They clearly worked in influenza, and they appear to work with seasonal coronaviruses as well. So what about your homemade face mask that we're recommending to the general population? Well, there may not be so great, depending on what you make them out of. If you have multiple layers of something thick, as opposed to a thin t-shirt, it probably works better. But there's also other data suggesting that some other materials are much more effective, including perhaps silk, if you want a fancy face mask. What about the study in the restaurant in Guangzhou? There's clearly much more than a meter between the index case over here and these people over at this other table. The group in Hong Kong, led by Hugo Lee, has been doing video analysis of the movements of every individual in this restaurant during the outbreak from the surveillance cameras that were present. And there's apparently no point when these people got close together. But why is the infection contained in this end of the room and not down here? There's multiple reasons. Another is that there was an air conditioning unit up here that was blowing this way, pushing aerosol down to that end of the room. The exhaust vents were closed off so that air was just being recirculated in the room. And so this analysis of this and looking at the tracer gas studies that show that aerosol was contained much more concentrated down here than in other parts of the room suggests that if the aerosol were contained in somewhat larger particles than the fine respirable fraction in the thoracic or inhalable fractions, that they might well have settled out before they got out of this end of the room, but there was enough air movement to keep them suspended. And that other people were not here as long and would not have gotten a very high exposure by aerosols. So while this doesn't prove that it was by coarse aerosols or that aerosols are a more important mode, especially in the homes where you may have lots of contact with other secretions. This does, along with the choir outbreaks and so forth, suggest that in the super shedding events outside of homes, aerosols may be a very important mode of transmission. Contact has always been suspected as being very important. The Metropole Hotel in Hong Kong, where the 9th floor accounted for all of the cases, it has been posited that maybe the elevator button was contaminated, and the room where the index case stayed. I understand 911 is considered a bad number, so the number was changed to 913. Different cultures have different ideas about what are bad numbers. That the contact may have been important. There haven't been much in the way of studies of air movement within this building. Similarly, in the Amoy Gardens, initially there was a lot of thought, because there were many cases in apartment block E, that elevator buttons and contact surfaces in that building may have been important, but a computational fluid dynamic analysis of the airflow between buildings and a compartmental model of airflow within this building matched nicely where the cases were in this outbreak. There weren't cases in these buildings that were not in the downwind plume of building E.
So
This was a case of aerosol from feces, yes. And
Dr. Milton, I'm sorry to interrupt. We would like to keep some time for question, and some people will have to leave at 2, so perhaps we should conclude here.
Okay, good. Well, you're going to talk more about other ways of protecting against and controlling air infection of various routes later. Certainly we want to get to understanding eventually how well do masks work for stopping COVID-19. And we are working on that. We are recruiting cases and studying them in the Gesundheit machine. So thank you for your time.
Thank you very much for a very interesting talk. So the floor is now open for questions. I know that Dr. Mar has to leave us very quickly. So if there is a question for Dr. Mar, I would appreciate that it be brought to the floor now. And there is already a question for Dr. Connelly. Does Dr. Connelly agree that healthcare worker providing close range care of COVID-infected patient would be inhaling small particles carrying the coronavirus, and would that pose an infectious hazard?
Sorry, just so I understand the question. The question was, would there be a hazard to small particles in the care of patients?
COVID-infected patient for healthcare worker providing prolonged care at close range.
There's always a risk for transmission and providing care at close range for prolonged periods of time, and that's why we recommend the personal protective equipment. So, and I had outlined that with the adequate training. I saw that in the chat box. We spent extensive amounts of time based on the previous literature that many of the transmissions from SARS coronavirus 1 were in settings in which there was not adequate training. The worst off were our physician staff because, doing 3 hand hygiene techniques to a doff, that's where most of the errors occur, was foreign to them. So we had to spend a lot of time on training education. So I think I would say that with adequate training and with the use of appropriate PPE, so the use of mask, gloves, gowns, goggles, or face shield in routine care, and then the use of N95 respirators if there's an aerosol generating procedure, I would feel that most healthcare workers should feel perfectly safe in providing the care. I've spent myself personally collecting all these specimens, hours and hours in the rooms, and I'm fine. I feel perfectly comfortable in the use of the PPE that has been recommended, but I take great care and attention to the doffing, because obviously if you make an error in doffing, you can self-inoculate yourself. Over.
So,
sorry, we have been asked if I would like to ask the presenters, could we summarize? We have 11 or 12 questions or more in the chat window that go back to Lindsay Marr's presentation. Are we able to create a Q&A, publish a Q&A out of the chat session due to the time that we're at this point? Can I get commitment from the presenters for that, possibly?
Sure, can do our best.
Happy to help with that.
Yes, if someone would like to compile or consolidate the questions and topics and direct us to answer certain ones, I'd be happy to.
Great. I will do that for everyone so that we will capture anything in the chat window. Appreciated. Sorry, go ahead.
Yep. So, right. So,
perhaps Raymond, are you thinking we're at that time? I didn't want to interrupt. If you had one more question you wanted to continue, or we can summarize the chat windows. Perhaps people should be writing them in the chat window, which we're saving at this point as well, to ensure we do revisit them.
Okay, so one question, would each presenter explain how they work with occupational health specialists like hygienists and what attention do they pay to their recommendation? Dr. Ma. Okay, I believe that she had to leave us. Dr. Connelly.
Thank you for the question. We work very closely with workplace health and safety and occupational health specialists. And in fact, we're the infection prevention and control individuals, the infectious disease epidemiologists work hand in hand with them in terms of the education and ensuring that we have adequate supplies of equipment, hand gel dispensers. And we also have used another group within our COVID unit response, and that is human factor specialists. So individuals that on the system level ensure that flow is appropriate. We try to ensure that the care delivery in the rooms and then the doffing process is done consistently. We use video analysis. We have an extensive website and also an app that we've developed that is a COVID app to be used as a digital assistant. So if someone were away for a couple of weeks and they were forgetting, so we did extensive amount of work with our occupational health people to be able to get the messaging across. And again, I'm happy to report we've not had a single healthcare worker transmission in 132 patients on our COVID units with the use of our procedures. Over.
So Dr. Milton? Okay. So another question here. The Sick Kids Hospital in Toronto released an opinion paper stating masks should not be used in school, and this can be perhaps also used conjointly with the comment that surgical masks are not considered PPE but as source control. So if you could please comment, Dr. Milton and then Dr. Connelly.
Well, I think I missed perhaps what the beginning is, the question is, but about schools, about use of masks in schools.
Yes, the Hospital for Sick Children advised against the use of masks in schools. So what is your reaction to this? And also someone put out the distinction that a surgical mask is a source control, not a protective equipment.
Well, I think that surgical masks definitely are a source control. And that has been the basis of the recommendation in the United States that people wear them because there are aerosols generated from breathing, but also from talking and singing. And the louder the talking, the more aerosol is generated. We know that there is a lot of virus in saliva, and this has been cultured from saliva, so that even if you think that the aerosol from the fine particles isn't an issue, these coarser particles clearly are, and you can reduce contamination with them. And I would strongly advise that they be used as source control in public environments, including schools.
Dr. Connelly.
I don't have much to add. I do agree with Dr. Milton about the source control and actually quote his paper, so thank you for that. The one thing just to remind you is that in children, the compliance with mask wearing in children from age 2 to 8 is about 15 to 50%. So this is a big issue that will have to be addressed, because you think about children, particularly in hot, humid environments. We've had people on the WHO calls from low to middle income countries around the equator. You won't get children to wear masks in hot, humid, sticky weather, and even at the best of times. So that's something that I'm not an expert. I'm not a pediatrician, but it is an issue that needs to be taken into consideration about children and mask wearing. And I don't have the answers, but the studies to date show very poor compliance, and you can understand why. Children with disabilities, autism, it's going to be, or developmentally challenged children, this is going to be a major, major issue. And perhaps that's why Sick Kids addressed that the way they did.
I think that it's also important to recognize that it's probably from middle school up that we are most concerned about transmission in schools because of the data suggesting that younger children, maybe before puberty, are less susceptible to the infection in households. So this may obtain in schools. We don't really know whether they're less contagious once infected, but that also could be true. So maybe we have a saving grace there. I don't know.
Dr. Milton, I had to cut you during your presentation, regrettably, because of time limitation, but several questions expressed interest in the use of UV light for air decontamination. Could you comment?
Well, there's data suggesting that upper room UV systems combined with good air mixing can produce very high rates of decontamination of air. In one paper, we were able to achieve the equivalent of 1,000 equivalent air changes per hour using vaccinia virus, which is very sensitive to UV. The sensitivity of coronaviruses is quite a bit less, but still actually somewhat more than influenza viruses. Our estimate is that you can achieve 100 or more air changes equivalent per hour with upper room UV systems. And they may, to the extent that aerosols are important in this, especially the finer particle aerosols, but also to some extent the ones like 10 to 20 micron that can move around quite a bit in indoor air, as Dr. Marr has shown, can be sterilized using UV systems.
So has there been any studies on the effectiveness of face shield without mask, just the face shield as a potential source control?
Yeah, I can address that. There are groups that are looking at the use of face shields as a source control, and particularly with autistic or developmentally challenged children who are not going to keep masks on. These are, and also in hearing impaired children, because remember, there's probably 500 million people in the world who, and about a third of them rely on lip reading. So certainly from the standpoint of large macroscopic spittle, it's going to actually reduce the potential for transmission from that setting if you were in close proximity. But it's not been as well studied because there are issues about, could smaller particles reach in underneath the mask. But certainly there was a major spray event from a sneeze or a cough, it would be a risk reducing measure. To what extent it would be does require further study. Compared to, say, a surgical mask or an N95 respirator. I just can't give you a quantitation about it, but I would say that the answer would be yes. And there are a number of groups who are looking at the use of face shields in some of these difficult to use mask population settings. Over.
And there is a paper from NIOSH here in the US looking at face shields as PPE with a cough machine and a lab and mannequins. But I haven't seen data on face shields as source control as opposed to as PPE. Over.
So I'm trying to find out questions because some questions are overlapping here. So
we did ask permission to continue. If people need to go, we will send the evaluations via email. So I can provide you additional questions, Raymond, should you like, from earlier, or I can...
I can stay 5 more minutes.
Great.
Okay. So yes, Kim, please. So here's an interesting question for both of you. Can droplets that land on the floor become airborne again?
You know, I'll take a first attempt at that. If you've got droplets, you know, that land on the floor, in all likelihood, there's going to be significant desiccation that's going to occur, and you know, unless people are picking up a product from the floor that hasn't been wiped up or cleaned and then self-inoculating themselves, the likelihood that they would become airborne and then inhaled would be, I think, and have replication and infection-competent virus would be extremely low, in my opinion. We do know that some of the RNA fragments that are found, and some groups have brought this up, when you find it in various places in the room, it's probably a reflection of cleaners and others, and they're just mixing around the RNA fragments in the room, and then they are picked up on the as a surrogate on some of the studies that have been done. But I think once it hits the floor, and if it's in a large droplet, you know, with the desiccation that's going to occur, the likelihood that you're going to be able to kick it up and then create an infectious aerosol would be extremely low likelihood, in my opinion.
So Dr. Connelly, you keep using this expression, like many of your colleagues, about RNA fragment. And as a virologist, this is something that I would like to explore a little further, because certainly you cannot refer to naked RNA, because we know that naked RNA is very, very rapidly destroyed in the environment. And anyone who has done wet laboratory molecular biology knows the extraordinary precaution that we take to preserve RNA and protect it from the omnipresent RNases. So if we detect RNA, that suggests to me that the virion particle is sufficiently intact to still preserve its RNA. So it cannot be a naked RNA fragment.
I'm using the term loosely, Raymond. And we do know that these are not live virus particles, and we know that from many studies that have been done where they're not able to find replicatable and infection-competent virus. And we find this in many of the patients after day 10, up to about 6 weeks, where you can get RT-PCR positivity, and there is no cultivatable virus. There's been at least 3 very well-done, elegant studies that have shown that.
Acknowledging, of course, that cultivating virus is not as sensitive as cultivating bacteria, and that the sensitivity for culture is much, much lower than the detection by PCR. So you might be hitting on the threshold there. So the reassurance is perhaps not 100%. Yeah.
But it does correlate with the rise of neutralizing antibodies. So I think that also is consistent evidence, and the fact that there are 3 independent laboratories from different parts of the world that have got quite consistent findings.
It's certainly the presence of antibody would be significant when we consider the persistent shedders, where the viral load in samples, though we can detect it for 30 days or more, we know that the viral load is very low, and if, as was the case with influenza, these viruses are coated with antibody, then certainly you're making a good case. But if we are talking about aerosolized particle in the air early in the infection, there are no antibodies at the time, and you have to acknowledge the limitation of culture in terms of sensitivity.
Yeah, I think we want to also acknowledge that, for example, in influenza virus, we know that different cell strains are quite different in their permissiveness for contemporary strains. And I think that you know, there's only now beginning to be work really looking at optimal strains for culture of SARS-CoV-2. So we may learn more about what the ratio of RNA copy number to actual infectious viruses as the virology and matures and the ability to culture these viruses matures. I know that Matt Freeman's lab and others are Ralph Barrick's lab and others are working on developing optimal strains for culture, and that may give us new information about what's really a competent particle and what's not. In flu, it does tend to look like you have 300 to 1,000 RNA copies generally per competent virus, at least with low passage MDCK cells. Even there's some suggestion, for example, with these engineered strains coming out of Yoshi Kawaoka's lab, that there you can find cell strains that are actually more representative of the actual airway epithelium and in their permissiveness for these particularly H3N2 strains. So I would take just a little bit of caution about interpreting results that are done with standard Vero cells.
A question for Dr. Connelly from the floor. When long-term care personal support workers spend extended periods in intimate range of suspected or confirmed patients in closed room with poor ventilation, why would respirators, meaning N95, not be endorsed?
Thanks very much for the question. I think in settings in which there have been outbreaks in long-term care facilities, and we had a number of them in our setting in southern Alberta. They were all wrestled under control without the use of N95 respirators. And I think one of the key issues was the training and the ability of the healthcare workers to understand the appropriate use of PPE. One of the things that's very important with long-term care is there's a great deal of care required for the feeding and then the turning of these elderly individuals. So self-inoculation vents become very important. So I think that's a very important item, is to look at the training and the expertise and the appropriate donning and doffing. And perhaps that's something that we didn't have the level of training and expertise amongst many of the healthcare workers in long-term care settings. I think the use of appropriate PPE and excellent hand hygiene is enough to be able to prevent transmission of SARS coronavirus 2 in the long-term care settings. We had at one time up to 30 long-term care outbreaks, but they've all been wrestled under control. And N95 respirators were not a component of the PPE. It was the use of medical masks. But again, I want to emphasize the training and the expertise in the donning and doffing is very important, that this be done appropriately, and that the healthcare workers understand the importance of the donning and doffing and the appropriate use of the PPE and excellence in hand hygiene at all times. If there's an aerosol generating procedure, for example, high flow oxygen that's humidified, absolutely we would recommend an N95 respirator, but otherwise not.
Dr. Milton, I was wondering if you could comment on amount, the volume of air that need to be sampled for meaningful detection of aerosolized virus.
Well, it's going to depend. The volume of air you need to sample is going to depend on the environment and the source strength of any cases in that environment. We saw in influenza that there was no correlation between how much virus we could detect in a nasopharyngeal swab and the amount we detected in their exhaled breath aerosols. And there was about a 10 to the 8th range of shedding into aerosols among influenza cases. So it's really hard to prescribe. It's going to depend on what your goal is in doing this sampling.